Hello, everyone. Welcome. We'll just give it a minute or so for other people to join, and then we'll jump on with the intros. Thank you. Okay, it is 12:01, so I will start. Welcome, everyone. Thank you for joining this month's Genomic AI Network webinar in collaboration with the Alan Turing Institute. This month, we are lucky to be joined by Joseph Halstead, who's the lead bioinformatician at Cardiff and Vale University Health Board. Joseph will be showing us his work on Variant Bank. This will be talking about the development as well as the implementation at the All Wales Medical Genomic Service into routine diagnostic workflows. There will be time for questions at the end. So if you have a question, please do put it in the Q&A tab. It should be right next to the chat tab on your Teams window, and you can upvote people in there as well for questions that you definitely want to be answered. So please do use that. And with that, I will hand over to you, Joseph. Thank you.
Hello, everyone. Thank you for the introduction. So I can skip this slide since I've just been introduced. But yeah, my name is Joseph Halstead, Head of Rare Disease Bioinformatics at All Wales Medical Genomic Service. So my day job basically involves managing a team that build pipelines and software that we use in the service to process all our genomic data. But also, I'm currently completing a PhD at the University of Manchester as part of my higher specialist scientist training, HST, looking at large language models and variant interpretation. So that's going to be the main focus of how we've been integrating those into the variant bank system, which I'll come on to. Yeah, so in this presentation, I'm going to try and give quite a lot of time to doing demos of the software rather than just going through slides, because I think it'll be a bit more interesting. But I am just going to go for a quick introduction to AWMGS, who we are, what we do, a bit of background, like on the problems we're actually trying to solve using AI, then get straight into the demo for how we're using large language models, AWMGS, and a bit about how they work and the proof that we're getting good results. And then maybe if we've got time at the end, a brief 5 minutes on other AI initiatives at the organization. So who are AWMGS? So we're All Wales Medical Genomic Service, and we provide genomic testing for the population of Wales. So that's across rare disease and cancer. We're located in North Cardiff at the CIGC site, so it's just off the M4, and we're part of this group called Genomic Partnership Wales, which includes Pathogen Genomics and Wales Gene Park, which are co-located at the same site. So we've got basically a lovely new building, a new site. We're trying to build like a whole life sciences hub. It's basically this area in North Cardiff. So yeah, my day job is bioinformatics of rare disease. So first of all, what is the problem that this presentation is about that we're trying to solve, basically? So this isn't unique to AWMGS. Any GLH in England or across the country basically is going to be hitting similar issues and trying to solve them in different ways. But we've all seen large increases in whole genome sequencing testing for rare disease. These tests return a large amount of genetic variants, which require interpretation. So a clinical scientist has to actually go through all that data, interpret it using their scientific knowledge, using these guidelines, which I'll come on to. And these scientists, they're in short supply, they take a long time to train. So it's created this bottleneck, basically. So we need to increase the productivity of analysis to make sure we can get more reports out in the same amount of time, essentially. So how can you increase productivity in like variant analysis? So there's this fundamental trade-off in rare disease variant analysis. Basically, you can make a much more sensitive test that has a higher diagnostic yield, but you end up having to analyze a lot more variants or go into a lot more detail in each variant. So for example, you can have very strict filters and you get really efficient analysis that doesn't take very long, but you miss loads of diagnoses because you're filtering out the variants. Or you could have very relaxed filters and you catch loads more diagnoses, but you can only get 1 or 2 reports out a week. So that's not very good. And basically, there's a trade-off here, and you can model this. There's this concept from economics called the Pareto frontier, where it's like the space that you can maximize a trade-off, essentially. So you could basically put your pipeline somewhere along this orange line here to have maybe at the bottom right, so it's strict filters, very efficient, but not much diagnostic yield, or at the top left, where it's really sensitive, but not very efficient. So each configuration is kind of a point in this space. But the idea is if you introduce a new technology, you can move this frontier outwards and basically get a test that's more sensitive and more efficient. So a lot of this presentation is going to be about how can we apply AI technology to basically move this frontier to have a more efficient and more sensitive test, which is what we want, because just moving it along that current line is just dealing with an inherent trade-off rather than actually innovating. So first of all, a quick introduction to variant analysis. So I'm sure most people on this presentation are at least a little bit familiar with this, or I'm sure in some cases very familiar, or experts. So when analyzing a specific genetic variant, so that's like a particular change in a gene that's been detected in the patient, they follow these specific guidelines, like the ACMG or ACGS 2024 guidelines. And these guidelines specify the evidence that should be collected and assessed, and how to combine these for a final classification. So you go away, you collect loads of evidence about the variant, and then unify that into a final classification. So you might say the variant's pathogenic, you might say it's benign or likely benign or VUS. And clinical scientists must also assess whether that specific gene is related to the patient's presentation or phenotype. So for example, if they've come in for intellectual disability, but you find something, the gene causes completely different symptoms in people that have a pathogenic variant in that gene, you can often just move on. You don't have to do the classification. So I'll go through these plots in a second, because I'm going to go through them actually when I do the demo as well. But these kind of frame the problem we've got a little bit. So this is the variant analysis increase. So we've got in the variant bank, we can keep track of how many variants we're having to analyze in each month. And you can see over time since 2022, when we started logging this, the amount of variants we're having to analyze each month is gradually going up. You can see a large spike here where we had a sprint to try and get rid of a backlog. But the point is, like, as we'd introduce more tests and the test change, do more whole genome sequencing, this analysis is increasing a lot, and we're not able to increase the number of scientists we have at quite the same pace. So, okay, you get more variants, but what if you classified each variant a bit faster? So this is the time taken to classify a variant, taken out the same database, so we record that. You can see there's quite noisy at the beginning, and then there's a gradual track down, and this is through essentially just applying traditional approaches. So a lot of it's just learning by doing. Clinical scientists have got more experience, they're able to do it faster. Some of it is by applying kind of a more proportional analysis and training and traditional management techniques, essentially, to increase productivity. But you can see we're still hovering around like the 2 to 4 minute mark. Some internal modeling basically shows we need to get it below 2 minutes to basically make it a bit more scalable in the future. Ideally, you want a case to be able to go from variants to report in under an hour. You could also just have stricter filters, right? So you're just looking at less variants per case. So that's remained relatively stable. So we're not actually being able to improve the filtering that much. You can see there's a slight trend down, but often the panels get bigger. We introduce new variant types, that kind of stuff. So it kind of counteracts that. So we're still analyzing maybe like 10 to 15 variants a case, depending on the test. So yeah, what we've done so far. So basically, to enable all this, we built this software called the Variant Bank, which is what the presentation is mainly going to be about. It's this software system for rare disease variant analysis. You can upload a VCF data, so basically all the variant calls we've got, filter those down. It brings in all those annotations that are really useful for scientists when they're doing the interpretation. Yeah, and they can record what they're actually the final results. Is this particular variant in this patient pathogenic? Is it benign? What codes do you use? What evidence do you use? We've also developed this ACMG automation package, which is like a custom Python package, and that goes and collects all the evidence for the ACMG, ACGS guidelines and suggests the code to the scientist to apply, which I'll come on to. So for anyone who's a software developer, Bioinformatician, just a bit of detail here, I guess. So it's a Django-based web application developed by us in-house. Implements variant filtering. There's got an internal frequency database, so you can track exactly how many times this variant's been seen before. Allows users to record their classifications and decisions about each case, and provides useful annotations for conducting variant analysis. So also got this ACMG automation package, which I just mentioned, and that goes and collects all the information. I'm going to give a demo of this on a Jupyter Notebook in a bit, so I'll just skip past that, to be honest. Right, so many rules in ACMG, ACGS analysis aren't amenable to rule-based automation. So in that package, right, we have some rules which are really easy to automate. So, for example, there's PP3, which goes, does a missense variant have a Revel score above 0.7? If it does, apply it. If it doesn't, don't apply it. Like, you don't need AI to do that. That's just a Python script, right? But a lot of the codes require interpretation of scientific literature, comparison to patient phenotype, understand the penetrance of the disease, other fuzzy reasoning and judgment, which make them difficult. You can't just write a Python script to do it. However, this kind of activity that large language models are really good at. So therefore, a lot of my research looks at the utility of LLMs in variant interpretation and the expansion of this existing software package to include LLMs. All right, so now I'm just going to get to a demo. So basically, just going to go through the variant bank, show you setting up some filtering, how we classify variants, go through the metrics, and then go through how we've actually implemented AI within it. Right, so when you come into the variant bank, you get a list of samples that require analysis. So, for example, here we've got a WGS case sitting here. If you wanted to do some filtering, click here. But for example, you can see all the information you might need about a patient. So you can see they've got this presentation. This is the family structure. So this is a trio. So you can see we've also got the mother and father and the sex and whether they're affected or not. You can see if someone's run some previous filtering workflows here. You can see no one's run anything for this one. And you just click create. You can select a virtual panel. So all the filtering criteria locked down for most of the scientists. I'm logged in as an admin, so I can change all the settings, but basically clinical scientists wouldn't need to change any of these, and then they just click create. And then they come back 10 minutes later and it's filtered. Once they've done that, they can view the variants that actually need classifying. So they can go through each variant and create new classifications. So if we find one, a test sample, so I don't mess up a live case. For example, we found these filtered variants in this sample. Let's say we want to classify this variant. We can select what ACMG code. So we can say this is a PVS1, so it's a loss of function variant, and let's say it's also absent in Nomad, for example, which is PM2. I'm going to say that will add up to pathogenic. They can record evidence, so and add comments for the scientists to see. And the idea is basically that this acts as our entire knowledge base for all the scientists to use and classify variants. So now to actually show you an actual case with AI in, so I just need to go back to the presentation, copy this URL. Right, so once the variant's finished, once the case is finished filtering, you get a list of variants like this. You click on the gene and you get lots more information that might be useful for understanding the gene. So it's what phenotypes it's related to. This is just all pulled from OMIM or similar. Links out to Nomad, various other websites that might be useful, such as Decipher. You can see if it's previously been classified in a lab, which is useful. The lab internal frequency, so you can see like this occurs, for example, this one occurs in about 1% of samples. So you can be quite clear that that's probably not going to be pathogenic. But then if you click on the actual variant, for example, this one, you get this information. So again, just some obvious links out to Nomad, got your splice AI values, ClinVar, Decipher, what the XMizer score was. But this is where my research was coming through. So this isn't actually a live case. This is one where I've ran the extra AI stuff that I'm going to go into. So here is where the ACMG automation package is doing its work. So it's deciding what does it think the automated ACMG classification is. So it's decided it's pathogenic. It's decided that it matches the phenotype. So this is using a large language model to compare the gene, in this case, which is NR2F2, with the patient's phenotype. So it says this patient's phenotype shows strong concordance with the associated disorder. Key matching features include, etc., etc. And it goes through and basically it understands the gene's variability, what the inheritance model, whether it's dominant or recessive, and compares that to the genotype in the patient. And basically uses AI to basically create this nice little summary and give a score between 0 and 10. So in this case, 7.5. It runs it. I'll come on to how it works, but it runs it a few times so you get a range. So you understand it's a bit variable. It searched the internet and found 2 papers that mention the variant. And then it's going through what codes it thinks apply. So it thinks PVS1 applies. So this is a rule-based one, just lots of Python code going through basically the guidelines. So it says it's a stop gained or frameshift variant in this coding exon. It's predicted to undergo nonsense mediated decay and a strong evidence from manual creation that loss of function variants in this gene causes disease. So it's applied it at PV. It's checked Nomad and seen it's absent and it's an area of good coverage. So it's applied PM2. It's checked ClinVar and found there's a couple of different submissions to ClinVar, and it's applied PS4 because it's also absent from Nomad. And this is where the AI stuff comes into. So it's gone, the patient is de novo and the variant is de novo in this patient and the phenotype is consistent with the gene, but also it's searched that literature, got the full text and gone, one study documented the same variant occurring as de novo in one patient, confirmed through parental testing in this one. And it says the same study reported another individual with an identical mutation, but parental testing was unavailable to confirm de novo status. And it gives the links to the papers so you can go and verify that's actually correct. As part of like the kind of governance stuff, we're going to always add an AI generated tag whenever we're using large language models to generate stuff. So the scientist knows that this is something that they need to go and check and verify themselves. So if we look at another variant in the same case, let's find one that's unrelated to the phenotype. Yeah, so this variant, it says it's a poor phenotype match, this gene OAT. This patient's phenotype shows minimal concordance with OAT-associated disease. Yeah, it just explains that basically this gene doesn't fit at all. And it's a try to apply some codes. For example, it thinks BP4 applies because it's a intronic or splice region variant, but there's no splice AI score, so it's applied this code. Let's see if we find one more. Here we go. So here's one it's predicted is benign. It's predicted it's completely unrelated to the phenotype. So the patient's phenotype shows no meaningful concordance. It's got a very low Revel score, so it's applied this. It's also too frequent in Nomad for this disease, and also flagged there's 8 homozygous individuals in Nomad. yeah, so that's basically what it looks like when you've integrated artificial intelligence into it. The metrics, I know that I showed you earlier, we can look at that. So time to classify by date. So we just got a little page that generates these those plots on the fly. So you can see this is basically the plot that was in the presentation. It is split out by different assay types, so you can look at just NGS or SNP array, for example. We have other things, so you can see it by what time of day people are doing the classifications, cases, how many cases each user is working on, how many classifications are being closed each date, that kind of stuff. But it's really useful for measuring the impact of changes we make to the workflow. For example, if we look at time to classify by date again, we actually introduced a change around April 2025, and you can see like the time went down quite a bit. So we did some extra training around this date, and then the time went down a little bit and then started creeping up again. So like you can kind of see like when you try and change something that it's having an effect, and you can track the effect over the long term. Right, so I think that's mainly the demo of the variant bank, and then I'm going to get basically back into some of the AI stuff, how it works, give some more detail, how it works, and some more interesting cases. Cool. So I'm just going to go for a few more examples of the ACMG automation package, basically classifying a variant. So this one in this gene MAGL2, that's a typo there. Basically, it's gone through, it's found all these references. It's decided these codes apply. For example, you can see it searched literature again and found 2 other studies documenting this variant as occurring de novo. It's also found some functional studies and flagged that to the user. So, for example, someone's done some immunocytochemistry on cells with this variant. So PS3 is like a functional study. There's this one where it's classified as VUS hot. It says the phenotype match is really good. Found one paper that mentions the variant. It's applied PP3 because there's in silico evidence that the variant affects splicing. It's found a functional study. So there's a paper where someone's done some RNA-seq analysis showing that this variant causes exon 16 skipping. And it's also found another paper, or the same paper, in fact, where someone has flagged that it's intrans or another pathogenic variant. Here's another one just with showing the description that it gives about the phenotype match. So how does this actually work? So we built, as part of the ACMG automation system, we basically added this AI module. So for the phenotype comparison bit, this is how it works. So it takes the patient's phenotype as human readable HBOs, the approximate age of the patient, which is important, and the genotype. And then it takes the gene ID, it goes to OMIM or MedGen, PubMed, panel app collects loads of information about the gene, like as much as it can, puts this in this gene facts file. We get a large language model to summarize that and create a gene report. Then we get another large language model to do a comparison between those two, so the patient phenotype and the gene summary, and then it outputs basically this thing in the black box here, but as a structured format, which we then display in the web application. We tend to use Claude 4.5 Sonnet for all this. We found it's the best. We did some initial testing, which I can't remember if I've got a slide on. So this is what the gene review looks like. So it literally lists all the different systems in the body, basically says, okay, this gene can affect this system, how, and give some notes. So for example, this particular feature might be very variable. it's part of this particular disorder, etc., where it's getting this information from, discusses stuff like the inheritance, the penetrance, the variability. So it's got this knowledge base then to compare to the patient. The phenotype analysis engine then goes through and compares that gene review to the patient information. So you can see a screenshot of it here, going through basically comparing all the patient information in a table versus that expected by the gene, going through it, thinking out loud so it's easy to debug. Yeah, and then it basically outputs a JSON file, which we can ingest and use programmatically. So the actual literature search bit, so that was the phenotype bit, but we actually go out and search literature, right, and find other papers that mention the variant and collect all that information. So if you just went to a large language model, if you opened ChatGPT or Claude and just said, gave it the variant and said, apply these codes, it would do a terrible job. It might hallucinate. The paper's probably not in the training data because it might be only published like last month, for example. Even when you get to go search the web, it doesn't have access to the kind of tools it needs to search for a variant properly, because lots of variants have different ways of there's lots of ways of representing the same variant, right? The HDVS can be different. You can use a different transcript. The gene symbol changes. You might use build 37 versus build 38. So basically, we need to ground the large language model, put a harness on it, so that it can do the job. So we get some code that searches for literature using specialized variant search engines such as LitVar, Variomes, and ClinVar. We collect the full text of these papers if possible. So we've got code that interacts with the APIs of all the main literature search, literature publishing, scientific organizations. We then give that full text to the large language model and apply the relevant, ask it to apply the relevant codes, explaining what the codes mean, what the limitations are, what it needs to consider when it's doing it. Finally, if there's multiple papers, we collect all this information together and produce a synthesis. So if there's conflicting evidence, for example. And here you can see just the prompt we use. So for example, you're an expert geneticist analyzing scientific literature to determine the applicability of specific ACMG codes. We give it what information it's going to get. So the article title, the abstract, the text, all the information about the variant it might need, and then we break down the task. So what does this package actually look like? So I'm actually just going to, some of you might be less interested in this than others, but basically if you ever use Jupyter Notebook, I'm actually just going to run through what it looks like using this package to collect all the codes about a variant. So it's basically all abstracted away. So we can just go from the package, import this object called variant classifier. This is probably shouldn't be doing a live demo. So if it fails, I will skip past it. So you create this classifier object and then you add the variant. So you're literally just saying there's a variant. You get the genomic coordinates and some information about the variant. So which gene it's in, what the HDVS is, for example. So we're going to add that variant. There's some configuration here, mainly around like the nomad codes. So just do that. And then we just click. You can load more than one variant. So you'll see it's quite slow doing this step, but it's just because it has to load loads of files in, but it only has to do this once. So you can see it's added, it's now annotated like the variant with Revel, alpha missense, nomad, information about the gene, loads of other information it needs like splice AI. And then we're just going to go my classifier, calculate codes. It's then going to go through all the variants that we've added to the classifier and calculate the codes. And I've put get online codes equals true just because we're accessing the internet, for example, searching for papers and using large language models behind APIs. So this normally takes about a minute, so I'll try and talk through it while it's doing it. And if it starts taking longer than that, I'll go back to the presentation and we'll come back to it. So this is something we just added. What I've been doing over the last week basically is professionalizing the software package a little bit. So adding logging, that kind of stuff. So what's it doing? So it said it's initialized the variant literature for this variant. It's found 7 unique references from all sources. So found 4 from LitVar, 7 from Variomes. It ranks the publications. For some well-known variants, you might get like 200 scientific papers. So it's really expensive and time consuming to then go and collect the full text and put them into a large language model. So you can set, like, just look at the top 10. So it just ranks basically by looking at the abstract, which it thinks is probably the most relevant if you've got more than a certain number. So it's now processing all 7 papers. So it's gone to pmc.com and attempted to retrieve the full text. You can set it to do the analysis multiple times on each paper just because there's a bit of variability. You might want to take the average. Because this is the development version, we're not doing that. It's just to make it a bit faster. So yeah, it then goes through all the papers. We're using Claude Sonnet 4.5 and collecting all the information. So this should take only a few more minutes, hopefully, and then you can view the results. But basically, when we showed you the web app, where were we? When I showed you the web app and this information was here, right? This is what it's doing behind the scenes, essentially. It's just displayed in a much more pretty method when I'm doing it. I will continue the presentation. We'll come back to that at the end. Yeah, so one thing at the moment is quite slow, but it's what we call embarrassingly parallel. So you can just analyze each variant separately, essentially. So it's not as slow as that in reality if you're doing 100 variants or something. Anyway, so actually proving it works. So we took 107 variants from 11 WGS cases, quite small verifications at the moment. Analyzed them with this package with the AI functionality turned on and off. So of those variants, this is the kind of the distribution of the phenotype score. So most it gave a below score to, which kind of makes sense. Most genes aren't related to the reason of the patient's disease. Some very high. So we then looked at, okay, what's the phenotype score of the variants that are actually reported as relevant and put on the patient's report? They were much higher. What was the phenotype score of the ones that weren't reported? Tend to be lower. You do get some that are high because they're relevant to the patient's phenotype, but they were benign, right? So they don't get reported because they're not pathogenic. So that makes sense. So it's shown it's actually predicting quite well that which variants are relevant. We actually just did a correlation study versus Xmiser as well. So looking at all the variants and just correlating how well it correlates with Xmiser. The ones that reported are shaded in orange. So you can see sometimes this one here at the bottom, Xmiser gives it a score of 0, or we gave it a score of 7. So it's actually performing better than Xmiser a lot of the time. And that's often because we're giving it stuff like the patient age, like if it's a baby, and the gene is associated with intellectual disability when they're older, the referring clinician is not going to have logged that, right, as like a HBO term that they've submitted. But Xmiser is not smart enough to take into account of that, but the large language model can actually think, oh, well, they won't have shown intellectual disability yet because they're so young. So adding literature search and analysis. So a bit of a smaller scale experiment here. So we took 30 reported pathogenic variants and ran through the package with and without the LLM literature analysis turned on. Without large language models, it managed to predict that 53% of these were likely pathogenic or pathogenic. With literature turned on and that analysis, that increased to 83%. The remaining 17% are ones where they had to go to MDT or something to collect extra information about the patient. So you wouldn't really expect it to be able to do that automatically, which is good. And you can see in about 13% of cases, it applied this code, 26% this code, 23% that code. So it's actually quite a lot of pathogenic variants. It's able to find extra evidence and apply it successfully. So what's still to do? So optimization of prompts for the phenotype analysis, inclusion of more sources for gene reviews. So there's stuff like there's like you can access almost all entire books on genes through the NCBI API. So we really want to get that information, include it in the gene report if we can. The literature analysis stuff just looks at the raw text, so it doesn't get stuff from the supplementary information or figures. So we found some papers where the information required to classify the variant is actually in a picture, for example, a plot. It doesn't look at that currently, but there's no reason why with a bit of coding we couldn't use a multimodal model or extract the text from the images, that kind of thing. So it's just probably a bit of hard coding for now. Use large language models to select the most relevant disorder where a gene is associated with more than one disorder. This often comes up where there's an autosomal dominant and autosomal recessive disorder associated with the gene, and we can't apply stuff like PM2 properly because of that, or BS1. And this is what I've been doing over the last week or so. So refactor the code to make it a bit more maintainable, better logging, tests, queue systems, so we don't overload the APIs that we call, for example. That's basically just grinding out some relatively boring technical work. So how do we validate the software? So the AI stuff I just showed you isn't live yet. We're currently working through how to actually make that live and how to validate software that uses artificial intelligence in a clinical laboratory, right? So it's easy to show you that demo. Actually rolling it out so that it's safe and clinical scientists can actually use it in an accredited laboratory is another much more difficult problem. So it's important to think risks-based approach, like you would normally. Just think what can go wrong. Okay, if that goes wrong, what guardrails do we have to make sure that's caught before a report goes out or some wrong information gets to a scientist? So we've designed our system to fail quite loudly. Like so the clinical scientist will get a warning saying it's not worked properly, basically, and we do some QC checks, that kind of stuff. Making sure we've got the right training, and then collecting all the other information you'd do, like if you're validating any other software or a new test, so reproducibility, sensitivity, specificity. There's also these useful UCAS recommendations, which we've worked through and done a risk assessment against. So we're hoping basically that we can use this technology to reduce the time that it takes for clinical scientists to do the analysis. We want to do some experiments to see, can we actually use it to filter variants? So if a variant is completely unrelated to the phenotype, can we filter it out? And that's obviously a much more riskier thing to do than just showing annotations. So that's going to take some very careful validation, but it's something we're starting to think about. How can we use this technology to increase productivity and maximize efficiency? So as you've seen, when I've just fired it up in the Jupyter Notebook, it is quite slow. But it is embarrassingly parallel, as they say. Like each variant can be processed separately, each paper could be processed separately in parallel. So we're going to maybe think about putting it on AWS. So our lab is currently transitioning our pipelines from on-prem servers to AWS, and we're kind of thinking, well, I've been thinking anyway, about how can we set this up behind an API that's scalable and just to basically abstract it a lot of ways. So you just send a variant to an API and you get all the results back. Just simplify a lot of the code, make it so we can process hundreds of variants at the same time, that kind of stuff. We need to work up the design, but this is where kind of see the architecture going. And anyone who follows AI a lot online or just involved in the community, I guess, will be hearing a lot about agentic systems. So the current system, Python package, making calls to LLMs, lots of hard-coded data collection tools, prompting, RAG, etc. For example, we process each paper separately and then run a function to summarize the findings, but I've coded that in a Python script that says go to get all the papers, then summarize the information, then synthesize it, then output a JSON, that kind of stuff. And I've started thinking recently that a cleaner and more comprehensive approach would just prompting an agentic system to go search the web and do it themselves. And then we develop a series of tools called model. They use this protocol called Model Controller Protocol that basically give it a system of tools that and APIs it can use. So we might have a bit of a refactor and think about how we'd go to do that in the future. Oh yes, this is just a bit on MCP, so Model Context Protocol. It's a way that you can get plug-in tools and data into large language models in a quite nice way. So we can basically get agentic system possibly to do some variant interpretation, but this is very much at the stage where we just want to run some initial experiments. So I think if we've got another few minutes, I'll just go through another AI initiative that we're doing at AWMGS. So we're kind of looking for opportunities to use AI across the genomics laboratory, really. So I've been thinking a bit about AI within electronic test request systems to support clinicians' requests. So we built this demo app. Let me just start it. Sorry. 889. Right. So we built this little demo app where a clinician could potentially make a request. So just to add a dummy patient. Right, so the idea is the clinician would be adding the patient, what test they want, example, what samples they're sending, and then big pain point at the moment is the HBO terms. So both the clinician, they have to go onto the HBO website, look up all these phenotype terms to describe the patient and give particular terms. And it's a pain point at the moment for us. Sometimes they're written on paper, they're not electronic, going back and forth, that kind of stuff. So basically, can we use AI to turn raw text into HBO terms? So if we just type some actually some spelling mistakes would be a good test of it. This is why it doesn't work. So you can just type in some raw text like that. And I'll go into a bit how this works, but we've come up with a reasonably novel method where we get a large language model to split that text up into what we call atomic phenotypes. So it splits it up into like the minimal phenotypes. So actually, if we look at the logs, should be able to see it doing it. So yeah, if we go here. There we go. So it splits it up. So large head, it thinks macrocephaly, intellectual disability, webbed toes. It then uses vector search against the HBOs to actually find the most relevant HBOs, and another large language model then selects the most appropriate. You can see there it's correctly identified macrocephaly. It's correctly identified that intellectual disability is negated, so does not have intellectual disability, and also correctly identified I assume is a clinical term for webbed toes. You can also add additional terms manually, so yeah, or remove them. So yeah, we're doing some experiments, just trying to find pain points throughout the genomics laboratory and essentially use AI to fix them. Now, where was I? Oh yeah, so this is just basically how that worked. Split it into atomic phenotypes. For each of these, we vectorize them, conduct a vector search against all HBO terms and their descriptions, returning the closest n matches, like 5, for example. A large language model then selects one or more of these as the most appropriate for each phenotype, and then with a validator, large language model then confirms each selected HBO term is supported from the original text. All right, I think that's it. So I think we've got time for questions.
Thank you so much, Joseph. That was a really interesting and insightful presentation, and it's great to see such a practical example of how these tools are being used
or enabled in the routine genomic workflow. We already have some questions coming through Q&A, so I'll go straight to that. So hopefully we'll have time to go through all of them. If not, I can send you after the webinar. So first one is from Alex. What's the ballpark for the amount of resource that was required to set up and maintain the variant bank, workforce and capital?
So in terms of workforce, we've got about 10 bioinformaticians on our team. A lot of the work was in the initial setup, which is about 5 years ago, and then we just basically built on it bit by bit. So we've got a team working on it. No one full time. Like we've got like 10 full-time bioinformaticians or so, but no one's one person's job to maintain it. We all just do bits and bobs. Where required, it runs on a web server, in-house web server, which is probably about 10 grand, that kind of thing. So it's not too much. The AI stuff, we use AWS Bedrock as our provider for access to large language models, but it's relatively cheap. Like per case, you're probably looking at less than £1 of compute or tokens.
Thank you. And next question is from Krina. Is variant bank open access?
It's not at the moment. No, it's closed source. We're kind of torn between kind of the idea of making it open access and kind of getting that collaboration, plus the certain intellectual property kind of worries we have, and also some regulatory stuff around like does that count as putting it on the market, in which case then there's some other kind of medical device regulations that kick in and that kind of stuff. So but it's something we'd look at and be hoping, if anyone is interested in collaborating on either this or the automation stuff, like please get in touch, because yeah, we'd be happy to. Like a lot of the automation stuff, like the stuff we want to do, is probably going to be quite difficult to do with our team in the NHS. So like we are thinking about like what would it look like as either an NHS-wide collaboration, because that's something we could do, or is it something that could be as part of like a spin-out company?
Thank you. The next question is from Poppy. What has the governance process been like for deploying the AI element? You mentioned including the AI tags. Have there been any other interesting insights or learnings?
Yeah, so we've managed to dodge a lot of the governance stuff by how we've structured the code, I guess. So for example, it's creating a summary of a gene using publicly accessible sources, and there's no PID there. It's comparing a list of patient phenotype terms such as intellectual disability, short stature. Again, that's not PID, that's just a not identifiable information. So there's no information going to the large language model, like leaving our servers that is anyway PID essentially, and individual variants aren't classed as PID as well. And we had a risk assessment and ran that past our information governance team, and they agreed. When we go into production and actually start running this and showing it to the clinical scientists routinely, we're going through essentially the traditional validation structure we would in the laboratory, just making sure everything's checked off, doing risk assessments for all the most risky parts. Our hospital does have like a Gen AI policy as well, which we're going to have to follow, which is basically just making sure that any use cases where we're using Gen AI to do something like this is flagged and put on a risk register, that kind of stuff.
Thank you. Next question is from Belma. How does the model deal with different nomenclatures in the articles?
So to find the variants, we use like these search engines, which are kind of like, they use lots of hard-coded rules to do that. So they convert the variant into every possible kind of nomenclature that might be used. So for example, if you're describing the protein change, it'll use the full amino acid change and the short amino acids terms, for example. That then finds the paper. What we then do is put as many of the different ways of representing that variant to the model. So we describe like all the different ways that variant is described, could be described, not all of them, but the most common ones, and then hopefully the larger language model is then able to match it up. We have run into some issues with that. It's mainly on the search though. The search just isn't perfect. For example, some people literally say at position, the papers that describe at position 362, a proline is changed to a valine or something like that, which just you can't search for very well. So we do miss some papers just because we can't find them, whereas the clinical scientists somehow manage to.
Thank you. And the next one is from Raymond. How can you ensure the papers it references are not hallucinated?
So we find the papers with the search engine and then put that paper into the context. So the paper that it's getting the information from is the key in a dictionary, basically. So when we just use that reference, it's not what the large language model is coming up with, it's from the search engine, if that makes any sense. But there is a risk that that there is hallucinations even with that grounding. We found if the grounding and putting the whole text of the paper into the context does tend to fix it, but it's why we had stuff like that tag that says this is AI generated. You must verify all the information. We're rolling out the training to make sure the scientists know that. But yeah, it's like hallucination is like a risk. But to be honest, on the frontier models with that grounding, it's pretty minimal. Like I haven't in all our testing we've done, I haven't seen a hallucination yet.
Thank you. And the next question is from Stephen. What is the default for the AI-generated phenotype match? Given the risk of hallucinating, will it notify the user about the need for more data, or will it always report low or high match for phenotype?
So I guess if I can go back to slide, was it? Oh, my screen's gone black. This one. Yeah, so it does display a range of max phenotype scores. There's a -1 option here, which it's using, which we've done for there is not enough information for it to make a decision. So in the prompt, we do say if there's no information about the gene, report that, and don't make something up, essentially. But again, the grounding with all that information about the gene. So searching PubMed, doing a literature review on PubMed automatically, getting the information from OMIM and Panel App, and putting that into the context tends to stop hallucination. But yeah, like it's fundamentally a risk of this technology, right? It still remains even with all the guardrails you put in place, which is why, for now, human in the loop is a must.
Thank you. I have a couple of more questions. So let's see if we can go for all of them. So from Nicholas, for the 17% that was not correctly classified of the 30 variants, what was the incorrect classification? Example, VUS called pathogenic?
So I think that test set was, let's go to that. So that test set was 30 reported pathogenic variants. This isn't a very large data set, which is the main limitation of this experiment. It's mainly because I want to speed up the software a bit and get my rate limits increased on the large language model APIs before I run larger experiments and work out all the bugs, because if it crashes halfway through, it's expensive to rerun it. But so that 17% that were not classified as likely pathogenic or pathogenic, they were just a VUS. They were classified as VUS, mainly hot VUS or warm. It just didn't manage to find, either it didn't find the paper that was required to get it to that limit, and the scientist did, or often the scientist went to an MDT and discussed it with the clinician, applied like one of the codes based on that. Or sometimes a gene is associated with 2 different disorders and we haven't built in the ability for it to disambiguify which disorder it is when it's applying some stuff like the nomad codes. So it defaults to not applying anything in those cases. So it doesn't apply PM2, for example, even when it should. So that's some further work, really.
Thank you. I think this is going to be the last question. Assuming, it's also from Nicholas, assuming literature behind a paywall is not accessible via LLM, if you do obtain the papers manually, can you add them to the LLM search?
We don't have that feature yet, but it's definitely something we could add. We did add like the ability to add API keys for stuff like Wiley and the other providers, but I found it was so buggy and it was maintaining it was quite difficult because it just changes, your API key expires. So at the moment we're just using if it's publicly accessible. I think in the future, like to try and find a way to access journals behind paywalls by either manually downloading or using API keys properly. But I only had an API key because I'm doing my PhD at Manchester Uni, and I'm not sure I'd need to carefully. So that'll expire eventually once I finish my PhD, and our lab doesn't have one, so it's not something probably going to look at for now. Okay. But sometimes the stuff like preprints is available even if the full paper isn't, so considering trying to do some search on bioRxiv, that kind of stuff as well.
Thank you. Thank you, Joseph, for taking the time to answer all these questions and for such a clear and practical session today. Thank you as well to everyone who joined and for the great questions. We really appreciate you taking time to be part of our Genomic AI Network community. We will share our recording on our website, and please keep an eye on the Genomic AI Network channels for details of upcoming webinars and events. So thank you again, and I hope you enjoy the rest of the day.
Thank you, everyone. Bye.